Okay, so we'll get started. So the goal for today is to cover RNA-seq expression. What is the technology? We'll not focus a lot on the technology. I'll just give you a brief preview of exactly what and how it works. And we'll focus mostly on methods, which is once you do this experiment, how do you quantify the data? What comes out of it? How do you think about analyzing it? How do you think about analyzing it? So we'll do the RNA-seq quantification today. On Friday, we'll do how do you analyze or do an exploratory data analysis with this kind of data. On Wednesday, which is next week, we'll talk about what's called differential expression, which is if you have a case and a control, how do you compare what happens, or how do you compare, or how do you do statistical tests to figure out what changed in a distributed way. And on Friday, we'll do a full-on hands-on, which will go from right from the beginning of quantification all the way to like the final data analysis. Okay. So your assignment one has been graded. You should have received it. You can go to Gradescope and check your comments and so on. If you have any questions, you're also allowed to ask for a regrading. Just one request. So when you're asking for a regrading, try to be polite. If this requires a lot of work from the TAs, it's actually a significant amount of effort to grade. Just imagine downloading or just running those 60 plus notebooks. It's not easy, right? I don't think there should be any mistake in evaluations, but you are the best judge. So if you think something has been graded, something has been graded incorrectly, you're welcome to raise a request, and there'll be an explanation for why the points have been graded. Please try to be polite. It requires significant amount of time. Okay, any questions about the assignments? Okay. So assignment 2 is already, assignment 3 is already up. So I think it'll be probably easier this time in terms of the time commitment and so on. There are just 2 theory questions and 1 implementation questions. And I hope you have fun with that implementation question. It's designed to have fun. Okay, so it should say lecture 9 and not lecture 8. So I'm going to correct it right now before we go. Okay, so we'll just to preview what we've been talking about and what we're going to talk about. So in one of the first lectures, I'd shown you this figure where we had motivated what the problem is about. So the idea behind doing RNA-seq is you want to quantify where exactly is the gene being expressed. And while this is a pure biological question, to answer this, and you will see, and hopefully you'll appreciate the technological advancements that we have to do to be able to measure gene expression. So last week we have been talking about DNA-seq, where we were just sequencing the genome. Now we are talking about sequencing the copies of mRNA or the isoforms that are expressed. Why do we care about it? Because DNA is very much similar across all different tissues. We have practically the same DNA in all our cells. But we have different proteins, and the reason we have different proteins is because we have different amount of copies of the mRNA, the mature RNA. Okay. So just to give you a preview of the scale of things that we're looking at and what mostly we'll focus on. So the transcriptome or the total RNA of the cell, there are different kinds of RNAs in the cell. There is pRNA, there is ribosomal RNA, and then there is mature RNA. Only 4% of the total RNA, which is your ribonucleic acids. So if you take a cell, you quantify it, only 4% of it will be coding for proteins. The remaining 96% is functional. So it carries out other purposes, which includes regulating the amount of mRNA you make. But otherwise, it's not really doing anything directly about synthesizing the proteins. Okay, not directly, but it tries to modulate the amount of what you end up coding for the proteins. This 96% has multiple different components. So it has a component about of rRNAs, which constitute around 90% of the functional part. Then there are tRNAs. So tRNAs play a role in, these are what will come and bring the anticodons when the synthesis is happening. So there are different kinds of this, and we're not going to focus so much on it. These are small nuclear RNAs, small nucleolar RNAs, miRNA, and so on. So people have different interests and they do study it, and I think they should be studied. But the focus for all practical purpose for this class will be on the RNA, which is the mRNA, excuse me, which codes for protein. Okay, so the biological goal is very simple. We want to quantify the number of mRNA molecules that we have in a cell or in a particular tissue. The reason we are doing this is because I have different tissues in my body. I have heart, kidney, lung, liver, and so on. They have exactly the same DNA, but the proteins that they're synthesizing is different. The reason they're synthesizing different proteins is because they have different they have different variety of mRNA molecules, and B, they have different quantity of the same mRNA molecules. And you'll see in a bit how exactly it is quantified. Before we do that, we have to talk about some biology before we actually talk about how do we actually capture it. So the biology starts in the nucleus. In the nucleus, your DNA will get synthesized into pre-mRNA. This is called pre-mRNA because it's not mature. This happens because your RNA polymerase will come. It will start to synthesize the RNA strand. This becomes one of the strands that eventually will get exported out of the nucleus. So this is your nucleus 4. And this is where the protein synthesis will begin. So there are two key steps before protein synthesis happens. One is your pre-mRNA gets converted to mRNA. So there are a number of things that need to get removed before you start synthesizing or using that particular mRNA to synthesize the proteins. And we'll just see in a bit what exactly happens. The entire process of going from pre-RNA to mRNA involves a different number of steps. But just for the simplification context, we'll just talk about one particular process, which is called splicing. So if you go back to lecture 1 or lecture 2, I had chalked out there are different processes in which you can regulate the final amount of protein, and we'll not talk about all of them. We'll just talk about one of them, and that too very briefly. So if you look at the proteins, the gene that is synthesizing the proteins, or the mRNA that's synthesizing the final protein, is going to be much, much longer. So if this is your protein, the green thing is your protein. This must have been synthesized from an mRNA molecule, which is going to be longer than the actual protein. Why is it longer? The reason it's longer is because we have different regions that will not be translated. There are two of these. There is 5' untranslated region. This is also called 5' UTR. And then there is 3' UTR. So this is 3' untranslated region. So these are sort of control centers for controlling which proteins will come and bind and how exactly the mRNA synthesis will happen or the protein synthesis will happen. This will not result in your final protein. You have a special start signal here, which is ATG in most cases, which is the start codon, which is where the protein synthesis will begin. Before that, you have the mRNA transcript, or you have a region which is called 5' UTR, but this is not going to result in amino acids. Similarly, at this step, you have the termination codon. Once the ribosome looks at it, it will stop synthesizing the polypeptide, and this will not the ribosome will kind of keep somewhat going further, but this will not result in a final protein sequence. Okay? So this is called the 3' untranslated region. So 5' UTR, 3' UTR, very, very important control centers. Not going to be part of the discussion for the rest of the talk, but just want you to be aware. This will make its way slightly later in the course when we start to talk about other different assays. Okay, so if you look at the gene itself is made up of different components. These are primarily exons and introns. Exons are the coding part, introns are the non-coding part in some sense. So this is what the gene looks like. This is your exon 1, this is your exon 2, these are your 5' UTR, these are your 3' UTR regions. Now, if you have different copies, different number of exon copies, so exon 1, exon 2, exon 3, and exon 4, what you see in the middle area is what are called introns. For it to synthesize proteins, these introns need to be excised out. Okay? So this is in this form called the premature RNA or pre-mRNA. The mRNA will have only exons. Okay? So this is what eventually will lead to the final protein sequence. And in the process, what will happen is these exons need to be selected. So there are multiple choices. It becomes a combinatorial problem at this step, right? So you have 4 of these exons. So in one possibility, you will have all 4 of them come together. You can have some of them come together, or you can have a combination of them come together. Okay? So this is what is called alternative splicing. So the splicing machinery, which you'll just see in a bit, what it will do is it will remove these introns and just join the exons together. Okay? So it's a very fascinating machinery in itself. It's able to carry out this in real time. Okay? So just to give you an example, it's an interesting example, which is from the same gene. So this is the calcitonin/CGRP gene. It has 6 exons. In the thyroid, the exon 1, 2, and 3 are taken together. So these become constant. The 4th one is excised. So the 4th one will come and join to exon 1, exon 2, exon 3. This will make the calcitonin gene, the calcitonin protein, calcitonin mRNA. Whereas in the nervous tissue, the same mRNA, the same copy of pre-mRNA will get excised slightly differently. You will retain the first 3 exons, but instead of exon 4, you have exon 5 and exon 6. And this results in an entirely new protein. So this is thyroid, this is nervous tissue. Same DNA sequence to start with, same pre-mRNA, getting spliced differently based on which exons make it to here. And that makes all the difference in the tissue. The one being nervous tissue, the other one being thyroid. Okay. Any questions about this? There's one more example of NTNG1, and there are multiple examples. These are cherry-picked in some fashion because these are also very well known in the field. Yeah.
Previous slide, what factor actually led to the selection of Hexin-4 in thyroid and identification? What is controlling all this?
Great question. So we'll touch upon this very briefly, but it's a question which I think the field is still sort of probing answers to. So in terms of what happens particularly is when I will show you the splicing machinery, it's a pattern recognition machine. That pattern recognition machine has the context about which tissue it is there in. It will recognize special patterns. For example, it could be 4 different bases coming from exon 4 in this case. It could be 4 different bases coming from exon 5 and exon 6. So in brief, there is a pattern recognition machine that's somehow able to recognize this. How exactly does it work? We know in some context, but we don't know in a lot of contexts. Especially the splicing machinery part, a lot of how the complexes work is a very, very active area of research. But also slightly harder because we still haven't been able to capture this properly, just because the technology is like just 10 years old in some fashion. Does that answer your question? Okay, so the other example is of NTNG, which has 10 exons. Exons 5 or 9 are spliced in and out. The remaining 4 are constant. It leads to 9 different kind of isoforms. So one thing which the technical part that I forgot to mention is what you're looking here are 10 different isoforms of the same gene. So this is sometimes called, people call it differently, like isoform is the canonical way of calling it. These are also, you can also think of this as 10 different transcripts coming from the same gene. These are all individually different, right? They're coming from the same sequence, the same set of sequences. They're coming from the same location in the genome, but they are still slightly different because they will eventually end up making slightly different proteins. So these are called isoforms. Okay? So one question you can think about is, if you even have a way to capture this, right? If you have in what happens is generally in tissues is you have different copies of these isoforms. So maybe some tissues will have exclusively copy 1A, but some tissues will have a mix of copy 1A and copy 1B. Okay. In most cases, you will have some exclusivity, which means that it will only express one particular kind of isoform, but nothing in biology is exact. So you'll always have one tissue where, for example, if I were to go back to this example, it's possible that some copies in the nervous tissue are still of this kind, but they'll be less in number. These will be overrepresented. So one thing that we also want to do is we want to be able to tell if we have a technology which can capture this data. We want not only the resolution to tell us where exactly in the genome it came from, so which gene is expressed, but also how many copies of the isoforms that particular tissue has. Okay? So it's a multifactorial problem in that sense, which is A, where exactly in the genome is the sequence arising from, and B, which kind of sequences are represented in your data. Okay, so how does splicing work? So I will touch upon this very briefly. Right? There is an intricate machinery. It's a good problem to work on. It's a very much open area computationally, and I think primarily also biologically, like what experiments we should do to figure out what sequence segregation happens and so on. We do know something about it, right? So the one thing that we know is the introns are GU rich towards their towards their beginning, and they have AG sequences towards the end. So this is, if you take the human genome and we know the human genome very well. We know which parts of this genome will get transcribed, which is what copies of the RNA will they make. We also know what are the exons and what are the introns in this sequence. We can use this data to tell us about the sequence characteristic differences between exons and introns. So this is potentially an exercise that you could do, and maybe we'll do this later in the class, is we'll take the human transcript and we'll look at the sequence characteristics of exons and introns. So one question is about the sequence characteristics that you can look at. The second question is about their size distribution, right? So you can see here, this intron is longer. Like you can look at these introns across the entire gene pool. So we have around 20,000, 22,000 protein coding genes. How many of these, what does the size distribution look like in terms of exons, introns, and let's say the 5' UTRs and 3' UTRs? Okay, so maybe I'll make this an exercise for you. It's easy to solve. But based on the data that we have, we can look at what these sequences are. So these sequences are GU rich and AG rich. There are also other introns which are AU rich and AC rich towards their end, but this is not the only possibility. Why am I focusing on these sequences? Because this is what the splicing machinery recognizes. The splicing machinery, remember what the job of the splicing machinery is. The job of the splicing machinery is to look for the beginning of introns and the ending of introns. What is it supposed to do? It's supposed to excise this out. It's supposed to remove this and then go back and join exon 1 and exon 2 in this case. Okay? Assume this is the simplest case, right? Only 2 exons. Machinery will come, recognize that there is a boundary because the exon sequences will have slightly different sequence structure as compared to the intron sequences. It will recognize this slide. This is called the donor site. This is called the acceptor site. Okay, so this is going to donate, this is going to accept, and you're going to essentially end up joining these sequences. Okay, yeah.
Does this happen always in a sequential manner?
In the sequential manner, as in one happens and then other.
Yeah,
You mean, okay, I see. Yeah, so it does happen in sequential manner. I think I haven't actually come across papers. Let me see. I haven't come across papers which show this, but from my brief recollection, it does happen. It does happen sequentially. But I'll check back and let me check back and get back to you. Okay. Alternative. So not always sequential. So there are cases where the last exon will get. Do you know how it jumps? Because the machinery has to jump all the way there.
is not sequential.
Okay, so it is not sequential forward. So I'll check back and get back. Okay, so in that case, I think it doesn't. Let's think about it. So in this case, the scanning doesn't actually have to begin from anywhere. So that way there is no sequentiality required, which is slightly different from the polymerase case or the ribosomal case, where you require a special signal to begin starting. So it's possible that the machinery can just recognize the sequence and that can happen asynchronously, and maybe that gets excised. But in some ways, it's able to come back and join together. So that's possible. So I'll check back and get back to you. Okay. Questions? Questions? Yeah. What if the ribosome goes? Okay, so that does happen sometimes. So the ribosome machinery will come actually after the splicing has begun, because the synthesis is happening in a different compartment, right? So we are still in the nucleus. To make the proteins, you have to you have to first get this copy to be exported out in the nucleus. There's no protein synthesis right now still happening, right? That is less of a likelihood just because the compartments are different. But what happens sometimes is, if the intron has somewhat a structure like exon, part of the intron can become sequence of your final mRNA, and that will end up making a protein which is actually imperfect. This does happen in certain times. Does that answer your question? So the ribosome is going to come later, right? At this point, it's not going to, because the compartments are. Oh, these are, so this part, the beginning part of these introns are zero. The end part are zero. Good question. Yeah, lot of sequences with you. So that is a good question. Right? It's somehow able to recognize like these four patterns rather. There is a lot of zeros in the sequence. So that's where the biochemistry kicks in, and that's where there is some special signaling that happens in this particular region that the spliceosome machinery is able to recognize, because GU is not very specific that fast, but it's somehow able to. So what is the exact mechanism? I think probably it's well understood. I don't know right now. I can go back, read it, and let you know if I come across papers. But for now, you can just stay with this idea that if there is some black box machinery which will come and remove these introns and then join these two exons. Okay, everyone, is that convincing enough? Okay. So the structure where this thing will happen is called the spliceosome, and the spliceosome machinery will come, sit here, recognize these sites, remove it, and come and then join again. These introns are then released. They can be recycled because you have these nucleotides that can be recycled. Okay, so question about what's sort of what Sabyasachi was asking, which is what happens if you have certain sequences of what happens if the ribosome comes, and what will happen. So the ribosome is in a separate compartment, but what does happen in terms of the errors is sometimes you have the wrong sequence. So it's a pattern recognition machine. Sometimes the patterns are misrecognized. So imagine you were to suppose have exons 1 and 2 by default in your protein, but it can end up excising 2 out. So you have exons 1 and 3 will end up making the wrong protein. The other case that can happen is the cryptic site selection, which again, I said there is a pattern in how these things are recognized. This pattern could be mimicked by different sequences in the exons or in the introns itself. So sometimes rather than excising the entire exon, you might just end up keeping some part of your, you might end up sequestering or removing some part of your exon, right? So this tends to conceal questions about what the pattern being very generic in that sense. So it is generic and these errors happen. But for now, we'll just focus on that there is a machinery which will come, remove your introns. Finally, you get your isoforms. These are all your normal isoforms. We want a way to be able to quantify that. Okay. What is the difference between? So this is one isoform of this gene. Okay. If it had been 1, 2, 3, that is also an isoform. If it had been 1 and 2, that's also an isoform. If it is 1, 2, and 3, that's also an isoform. If it is 1 and 3, that's also an isoform. So an isoform is the permutation combination that's synthesized in terms of the exon set. Yeah, so it's possible. Yeah, obviously. So all the possibilities that I talked about, it's possible that we never see a likelihood of 1 and 3 ever occurring together. So there are three theoretical possibilities, right? So three things to choose from. You can end up also making a protein with 1 exons, 2 exons, 3 exons, becomes a combinatorial problem, right? So this is the combinatorics acting here. But none, all the possibilities that you get, right? So 3 choose 1, plus 3 choose 2, plus 3 choose 3. Not all of them will end up making A. First of all, not all of them will end up making protein. B, not all of them need to make protein. And C, not all of them will you find in the entire human body. Okay, so there is some, maybe some of them have a different codon structure, which is not optimized for what you have or require in the body. So these are theoretical possibilities, not necessary that you'll see all of them, right? But computationally, we don't know, right? We don't know computationally which of these isoforms are there in the data before you actually start to look at the data, right? So there is some hidden part here, right? What we do know, because we have been able to sequence the human genome, we have been able to look at the RNA, is we know what are the likelihood or what is the possibility of these isoforms. So we do have this information available as to what are the possible isoforms in humans. Okay. Does that make sense? Okay. Mhm. It is related towards that part. Yes. Sure. So it doesn't make so as any machine, it doesn't make sense for that machine to make mistakes. But this is all under the assumptions that the entire environment is exactly the same all throughout time, right? Which will not happen if I indeed start inducing changes in the microenvironment. This could be in the form of mutations. This could be in the form of radiation. This could be in the form of stress, right? So if all these different factors which are playing a role, so imagine you have a machinery, but that machinery also requires presence of other things for it to be able to function properly, right? So it requires the proper flow of ions, it requires the proper flow of certain proteins which are going to come and bind, and they occur in different contexts in certain default manner. If that changes, the rest of the machinery will also start to change and incur errors in the process. So we can't incorporate the factors about what went wrong, right? We don't know unless you do go and do experiments. We'll not know what exactly went wrong, so that you're getting, you're getting this isoform versus this isoform. Okay, what is the mechanism that's hard from a sequencing experiment to figure out. What I'm saying is, I take my heart, okay? I take my kidney. I have no idea which isoforms are there in my heart and which isoforms are there in my kidney. If I take them and do a sequencing experiment, I know that their DNA is going to be very, very simple, but I have no idea what their RNA is. I want to know, I can figure out, I can do the DNA sequencing like experiment, and I can figure out which genes have how many copies of the mRNA. Because if I take that read, it will go and align to the gene. What I also want to know is which isoforms are there. Okay. That is the problem. So we'll touch upon this in a bit. But does that make sense? So let's assume I get cancer, right? Which is very, very likely at some point I'll get cancer. So if that does happen, I want to figure out at which step or what is exactly the thing that went wrong in that. Right? So there are multiple ways to look at it. One is there is some mutation that happened. One is I start synthesizing proteins which are not the right proteins. Why is it happening? It's happening because I now have a new isoform that my cell is synthesizing, which it was not supposed to. And that's why I'm getting a cancer and only that particular. That's not right. Does that make sense? Okay. Okay, so how do we quantify mRNA? Right. So this is the question that we are asking. So I'll stay away from there because it starts to echo. Any ideas? So you know about DNA sequencing, you know there's a magical machine that does this beautiful reaction, which I'll again, just for the sake of clarity, I'll draw it again because I find it fascinating. So you have your flow cell. This is something which is coming from your genome. Instead of this genome, I now want this to represent my mRNA. We have access to this technology that given a particular fragment, it will tell me how many copies of what is the actual sequence that I have there. So what would you have to do actually? Like assuming you have an ability to sequence the DNA and you want to sequence mRNA and you want to do it in a quantitative fashion, which is that you want to retain the information about how many copies of each gene, right? So when I'm talking about copies, what I'm talking about is this is D1, this is D2. Their DNA is N equal to 1, or actually N equal to 2, right? We have two copies of the DNA. So DNA is N equal to 2 because we are deployed. So if you look at the DNA sequence of these two genes, there will be N equal to 2 for both of them. But when we talk about RNA, this could have 4 copies. This could have 2 copies. Okay? Because this copies, how many number of copies of mRNA you have, that's a function of what is the level of proteins in some fashion. So the amount of protein that each gene will make is going to be, in the absolute manner, is going to be different. Same DNA, different copies of mRNA leading to different abundance of proteins. Does that make sense? Okay. So you were able to sequence this part. You're able to sequence this, right? I can sequence this. I want to sequence the RNA. How do you do it? Do we even need to you know how it's getting translated? So you know how it is getting translated, but you want to measure. So ideally, you want to just go and measure the proteins. So I'm going to give you my biased opinion, which is that quantifying these proteins is a hard problem. And there are technologies to do it, but it's still not there yet. Not there in the throughput fashion. Huge noise problem. So we can't work with the proteins. What is the next level of information that we can get? Is the RNA. The assumption there being this fact that RNA protein are correlated. This doesn't happen in reality, but the assumption is ideal thing that I would want to measure is the, I measure the for each gene how much protein am I using. Very, very hard problem. Technologies exist, they work, but they do not work at the scale at which we ideally would want them to work. Okay. Okay, so you will go from this direction to this direction. How do you do it? Yeah. The whole of this RT, the goes transportation. Just like polymerase, just like DNA polymerase, it goes from DNA to DNA. There is an RT polymerase, reverse transcriptase. You take the RNA copy, convert it back to DNA, you can throw this back onto your sequencer, you have your sequences. Okay, that is exactly what the technology called RNA-seq tries to do. The only step that it tries to be a bit smart about is if you do this for all the copies of your RNA, you will end up getting huge number of things that you do not require. You'll get extreme amount of ribosomal RNA. So mRNA is around 4%. And your RRNA is around, for all particular reasons, it's called 96. It's around 90%. If you do this generally, you'll end up getting like a lot of RRNA, which is somewhat interesting, but in most cases, you're not really, don't really care about it. So the innovation in coming up with this technology was how do you focus on? How do we focus on the mRNA? What is the way to focus on it? So mRNAs have a very special, very special signal, which is they have a poly A tail. Which means they have different number of A's towards the end of the sequence, towards the 3' end. And we have a mechanism to tap all these sequences which are rich towards their poly A. We'll not go into how exactly that is done. But again, there is a black box that exists, which allows you to profile all the copies in your cell which have a poly A tail. You can separate these out. You can convert them back to DNA. You can generate what's called cDNA. So this is RNA. Reverse transcribe it. You get cDNA. Put it on the sequencer. The same stuff that you did previously. Shear them before you do that, and you have your data coming out. If you tell you for each gene, once I get this data, I can map this. It will tell me which gene has how many copies, okay, or how many exons. So now the quantification will happen at the exon level, right? So because all of this data is assumed that it's coming from exons. How many exons are there in your sample, or how many copies of each exon are there in the sample? Is the problem clear? Okay, very good. So very briefly, this technology, what it's trying to do, it's a mix of in vivo, in vitro, and so on. This is a paper. This is an image taken from a paper which came out, I think, 6-7 years ago. This is actually also the default image on RNA-seq on Wikipedia, which is very interesting. So the way you start with, and this is just to help you visualize what exactly is happening. So you have this copy of your DNA. You have pre-mRNA, which is that you have exons, introns, exon, intron, so on. This is your pre-mature RNA. Intron, a black box will come, machinery will come, get it sliced out, get recognized introns, you get a mature copy of RNA. They also have poly A tail, right? So this is how you recognize that these are mature RNAs. You can fragment it, convert it to cDNA, or convert it into DNA, or convert it into complementary DNA. Place, get these sequences, do the exact same thing that you did with your previous data, which is that you align. Okay, clear? Technology is clear? Yeah. Okay, yeah, you're going one step ahead. Yes, so people have very good thought. So people have done this in the form of looking at real-time synthesis of mRNAs. So that's where the idea of having a fluorophore incorporation is used. So you can track the incorporation of U's in your data, which is complementary to your T's, and that will tell you at what rate, what is the kinetics of the mRNA being synthesized, at what rate is it being synthesized, at what rate it is degrading. So I don't have that part of the component covered in the class. My bias view is with that technology is still not there yet. It's good. It's a good starting point, but you're not there yet. So if anywhere I felt that we're not there yet, will not talk about that particular technology, but we are happy to discuss it later. Okay. So what does the workflow look like? So the workflow, this is a very biased view of the workflow because it has all the name of these tools that you will eventually, some of these tools you will eventually start to use. Once you get your FastCube data from here, yeah. Yes. Okay, good questions. Why do we fragment RNA rather than fragmenting the cDNA? The primers will anyway going to get made for, yeah, exactly. So the primers are going to get deposited here, not to the entire. In this case, it would not matter. You could actually shear it, but it's easier for the RT to be more efficient if you fragment it rather than the same thing, right? The polymerase, you're going slightly ahead. So barcodes, by barcodes, do you mean PCR handles? Yeah. So barcodes are different otherwise. Are talking about single cell stuff or no? Okay. So yeah, the PCR barcodes, right? So the PCR barcodes will be added towards this end. The reason you're doing the RT first is this is going to, the fragmentation first is because the RT is more efficient this way. It's the same mechanism whether you have to synthesize the entire DNA all over again. The DNA polymerase will start to veer off at some point. So it's not exactly the same, especially because this is happening in vitro in some fashion. So you shear it first and then you do this. Okay, is that reasoning clear? Very, very good question, right? Very, very good question. So the question is, okay, you are doing this RT, right? So when you do the RT will take care of the quantification. So the more copies that you have of the RNA sequence, more copies of the corresponding cDNA you will synthesize. Like that's an exact mechanism, more or less an exact mechanism, right? So in terms of, so this is your RNA, your cDNA. It won't be a 45-degree line, but it would be very, 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 very good. Everything will scale up according to the how many copies you find. Okay, very good question again. So it does matter to some extent. It doesn't show up here, but it will start to. We have to feature that part in when we start talking about the part. Yes. So very good point, right? So to think about this as a length problem. So just because, so think about this from this point, right? If I were to sequence this DNA, this is DNA1, this is DNA2. When I put this on the flow cell, right? These are of different lengths. Now, how many copies? So imagine when you run the sequencer, you have a choice of how many number of cycles of PCR you want in a fashion. And let's say, so by cycles, I mean cycles of PCR, which is the bridge amplification part. So let's say I do 5 cycles for this and 5 cycles. So in some ways, I'm going to sort of synthesize similar amount of, both of them will be amplified in a similar fashion. But when I get the dairy data, so you can imagine the final output is going to be comparable. But what that essentially means is for just because of the fact that I get more lengthier version of this sequence, its final quantification will be slightly larger to begin with. Because when I started with this entire DNA, I had more copies of things to amplify to begin with. I'm doing the same number of PCR cycles or not. So the final read count, right? So the RC read count for D1 would be, let's say, 15. Let's say this is 1.5 and this is 1. So the length is 1.5, this is length 1. If this is 15, D2 will have 10. Okay, does that make sense? Same number of duplication cycles. This is full length of your DNA. You start with more copies here to begin with, right? Because when you shear it, this will make, let's say, 3 copies. This will make only 1 copy. This gets amplified with the same number of copies. You will end up with slightly different copies. This happens in the genome world, in the transcriptome world. In the DNA world, it doesn't matter because when you start with the DNA, you only care about the entire DNA sequence. But in RNA, you have different length of the genes, right? Maybe we should do this as one of the exercises, which is let's try to plot the distribution of the length of exons or the length of the total length of genes. So you'll recognize that there's a huge spectrum of how lengthy our genes are. Anyone knows what the size distribution looks like? Size distribution of genes, smallest gene, largest gene. Median size of the genes. People. What's the size? Median size of the gene. Okay. Okay. I'm asking people, assuming you have had some exposure to it, but yeah, it's not a trick question, and neither it's a question to ask whether you know it or not. I'm just curious. So the distribution is, I think, if I remember it, I don't remember it, that's the problem, but I think it's around 2 kb is where it peaks at. That's the, I think that's the size of the exons. Anyway, you'll do this as an exercise. So whatever I'm saying, you don't have to take it at face value. So you'll see what the distribution of these exons and introns look like.
Are we only sequencing one mRNA?
No, you're sequencing the entire pool of mRNAs all at once. So you take that shearing thing, you shear it, you convert it to cDNA, throw this off on a sequencer, data comes out. That data is all the copies of 20,000 genes. So, okay, that's also a good question. And I think that's not there in the slide. Yes. This one?
No.
This one? Okay. So what does the data look like, right? So the data looks like once you get out of the data is, this is gene 1, gene 2, gene 3, 20,000. Again, it's not either the exact number, nor does it matter. Then you sequence one sample, right? So let's call it S1. So this is a vector, it's a column vector. This is 5 copies, this is 10 copies, 15 copies, 0 copies, 1 copy. So it's a vector which quantifies how many copies. So this is.
Do we know this vector before?
No, you know this after. We're just exactly going in that direction. How do you figure out what is vector? So this is at the gene level. This is at the gene level, which is good, which is a good starting point. You want to know which gene is making how much of a protein. But sometimes, in certain cases, particularly in diseases, you also want to know is you also want to know which isoforms change. Okay, so this is gene 1, isoform 1, gene 1, isoform 2, and gene 1, isoform 3. Gene 2, isoform, only one isoform, and gene 3, only. Okay, so here, so you saw that gene 1 has 5 counts. Now, among those 5 counts, this distribution could look like this. Isoform 1 can have 2 counts, isoform 2 also has 2 counts, and isoform 3 has. This is all theoretical, can be any permutation of. Similarly, for gene 2I1, it's only one isoform, so extend gene 3 is I1, I2, so 5 and 10. Okay. Good question. How after sequencing do we get as a gene or do we get as an isoform? The choice is in your hands. You choose what you want to quantify. The ideal way, and the more sort of granular way is to go from here to here. Okay. If you know this is just the sum. If you know only this, without having more information, you cannot directly convert this to this format. Why? because I know which isoforms should exist, but I haven't quantified them. I haven't looked at that. When I was doing the alignment, I was only looking at the gene coordinates. So I know where exactly that read went, right? So just to give you an example, this is gene 1. Its isoform 1 looks like this. Its isoform 2 looks like this. And its isoform 3, this is slightly an extreme scenario, but this is what. So when you do the alignment, you have a choice. You say, okay, is my read falling in these boundaries? If it is, I quantify the gene. Whereas here, you have to do some interesting math. This gets very, very interesting. I do want to cover it, and I think you should be able to cover it. So now what is the problem is I'm slightly jumping ahead, but I think that's okay. You have these 3 different isoforms. Okay. Now I take a read. This is my R, this is my read. How do you decide which isoform should it be? Gene, it is okay. Gene, I know these are the boundaries. It should obviously belong to this gene. No issues there. Isoform R. So think for a bit, and let me move along, and I'll answer this question in a bit. The problem is clear. Read. I know the boundaries of the gene. I know it belongs there. Isoform. I know there are 3 different isoforms. They have overlapping sequence. This exon is common. This is E, E, E. All 3 isoforms share 1 exon. Exactly the same exon. This read belongs, let's say, in this boundary. How do you assign it to one of these partitions? That's the problem that we're talking about. Okay. So why is it slightly different? Why is the mapping slightly different? Is in DNA sequencing, we could take the entire DNA, we could make it a Burrows-Wheeler transform, and we can figure out which gene is mapping to which location of the DNA. So this helps us figure out what is constant or what is there in my genome versus what I have in my data. And then you can use this for finding out mutations, which is what we did in the previous hands-on session. In the RNA world, right, the problem is slightly complicated, and that's why it requires more information. Is if I start mapping everything to the genome is a continuous body, which is made up of exons, introns, exons. When you get the gene, so this is your exon. When you get the gene, if you do this DNA sequencing, whatever is here doesn't matter, you're going to get contiguous sequences. Your fragments are going to be contiguous, right? But when I do the RNA work, right? When I do RNA, these things would have been joined together. Okay. So the read that I get, it's possible. This is your entire sequence of the read. It's quite possible that this half came from this half and the other half came from this other. Okay. So one read, one DNA sequence, how do you figure out where exactly it came from this particular location? Like that's the main problem that we have to be aware of. So this is this broad category of methods that try to solve this problem is called slice-aware alignment. Your alignment strategy will be different than doing a DNA alignment because you'll have to be aware that this read could have originated from a splicing machinery, which is that it would have come from 2 different exons, which are far apart. They're far apart, and you don't exactly even know which isoform they belong. Okay, is the problem clear? So this read that you're seeing came from this part, but all you have access to is the DNA sequence data. So how do you actually think about how should you do this kind of alignment? Where should you draw the partitions? Okay, so this just quantifies, and this you have seen this version of the picture in my motivation, is how do you assign each read to an isoform unambiguously, right? So, for example, you have you have access to a splice aware alignment. So it takes your read, it can split it if required to map it to the genome, because the transcriptome is spliced, or the transcriptome is a subset of the genome. Once you do that, you want to also figure out which isoform these reads are coming. That's the problem that we're going to deal. So there are multiple solutions to this problem. This figure just quantifies slightly more, flushes out that idea a bit more. How do you map it, right? So the first idea of mapping is, you can think of, so we know the DNA sequence of the human. We actually also know what the transcriptome generally looks like. So we have some idea about what all isoforms are expressed in different tissues. And we have a collection or a superset of these isoforms that we have access to. Okay. So in the last lecture, we had looked at the entire DNA sequence. So you downloaded this. You can also download a collection of all these isoforms of features. This is also available probably. How this collection was formed? Very, very good question. This is 15 years of work overall, basically. So Ensemble and NCBI. So Ensemble is the European Nucleotide Archive website. So you look at NCBI first, right? Like that's the National Center for Biological Information, NCBI. That's the US entity, and then there is a European entity, and both of these sort of talk to each other, and both of them think they are more supreme than the other. But the idea what they did is they looked at like multiple different donors. So they took RNA-seq. They took patient 1, patient 2, patient 3, did RNA-seq. So they did RNA-seq for all of these. And then they did what's called de novo assembly, which is you start from the RNA sequence and you start to put together what the isoform structure would look like. So if you remember, I had talked about a graph kind of a structure, which is, let's say you have this read coming, ATCG, and the next read is, let's say, ATCVD. You know that these share a portion of the sequences together. Okay, so I can put these two all together and I can make that, okay, I have this data coming. This is obviously oversimplified. This means that I do have an isoform whose sequence will be ATCGP. Does that make sense? So you can use this data. So I think this is my highlighting here. This sequence clear. Okay. So AT, actually, let me flush this out. Okay. So this sequence, this is the sequence of the read. ATCG. This is read 1. This is read X. Doesn't matter what. This is the read number, not the R1 or okay. The second, the read here is TCGP. Okay. This portion, which is TCG, that is an overlap. It overlaps, right? So I can collapse them together and I can say that there is definitely, with all extreme simplification, there is definitely a sequence in my RNA, which is like this. Good question. How to make sure what is it is that is not random. So the graph is able to builds upon this idea. It takes into account A, what is the error that you're calculating. B, this is not actually single end data, so it's actually pair end data. I'm simplifying it, and that's why it's much. It looks like you will get a lot of errors. But when you start to talk about pair end sequencing, that is very, or not very, but a lot more exact. So so if you take R1, this is your corresponding R2. Now, this is one read pair. You take another read pair which has this portion, this is an overlap. Okay? This is R1 prime, and this is, let's say, R2 prime. If you see that this portion also overlaps, then you can put these two together, and then you have that one fragment of the sequence. So it's harder to do this with single cell, with single end. It's doable. It won't be that exact, but if you have paired data, this becomes like very. So we'll not do that exercise. It's slightly complicated. Like you have to talk about graph structures. So that's why I said in the beginning that I'll not talk about assembly problems. They're actually very good problems, but slightly harder to handle in this course, just because we have to slightly change the trajectory of things that we're talking about. So I'll not talk about it, but that's the general idea. The general idea is so it's regularly updated, which means that this is bootstrap in a fashion, right? So you start with like 10 different donors who submitted their data. We ran these kind of algorithms. We came up with one possible structure of what isoform. So this you're making a database. You're making a database of the isoform. This is isoform 1, isoform 2, isoform 3. So okay. Now this is from 10 different patients. Now tomorrow, 15 more patients come from India. Let's say. This is European, so you have this data coming from Europe. And then now 20 different donors come from India. You will do the exact same exercise and find out which of these overlap. Okay. There will be things which will obviously overlap because we are all humans in the end, but then we are of a slightly different ancestry. So we'll also have additions of I1, I2, this is coming from a different model. This keeps on expanding. So one of the problems that people who work in cancer, and I keep talking about cancer because that's sort of one of the most studied diseases, is they look at when you start from this isoform database. So you will do this exercise. You will take an isoform database as in like a set of sequences from the isoforms and map your data. You can also find out, are there novel isoforms in your data? And this happens a lot in cancer. Right? You have a particularly misspliced organic isoform which has not been characterized, and it's not been characterized because that particular disease is special, that particular patient is special, cancer is complicated, cancer is very individualistic, so it never was that isoform ever quantified. You can use that information to build or to identify a novel isoform. So I'll talk about one of the papers that try to do that in a different setting, but yeah, these are the questions. Yeah. How are we identifying a new isoform? So what we do is, again, we take our data. So we take the paired end data that we have. So I did an experiment. Again, I take my kidney, like my kidney has some disease. I will take the data. I'll do RNA-seq on that kidney. I will do a de novo assembly. So I'll start to put this data together. And then I'll go and match it to the database that I have. How many of these isoforms that I'm characterizing in my data? For this, the data needs to be like super good quality. You need to sequence a lot more because this is not a problem to assemble. And then you can figure out which isoform is new, which was not characterized previously. So it's just a matching problem, a set difference problem in that sense. This is a set. So this is set S1. You get a set S2. What are you looking for? If you want to quantify novel things, data science. So set one is the characterized. So this is characterized isoform. Okay. In the set configuration, this is set one, set two. You're looking for what is sort of there in one versus the other, right? So this is the difference on both sides in some way. So what is there and what is not there also you can characterize it. Is that clear? If we are going for differential, okay. So if you're doing differential, which is you're interested in what happened in case versus what happened in control, you're generally don't care about, generally you should not care about the novel things because you're caring key what existed and what has changed, right? So that is the differential part. This is so it could happen exactly. So again, it depends on the biological question or biological hypothesis you have. Do you think it is happening because something new is happening, or you think it's happening because something was already there but it's in the different quantity and that's what is making a difference? So that is a different question to ask. You can do both. I think the de novo is great. I don't use it generally because I don't go to those kind of data sets, but you know, if you have to, you can do it. It's doable. You should do it if you have a particular question in mind. Okay, so we have 20 minutes and let's see how to best structure it. So okay. So let me talk about the methods for quantification. And these are, I think, we might not be able to finish everything. Actually, let me come back to the methods a bit later. Let me just start with the motivation. So I'll touch upon the methods in the next class before going to quantification, before going to the matrix. So there are three different categories of methods which try to solve this problem. Okay, so right now we'll assume that these are black boxes. These methods will give you data, which is this vector information, which gene or which isoform has how many towns. Okay. Now, the essence. So the gene problem, the three methods try to solve the gene problem slightly differently. But assume you have this information available. You have this information available that this is the distribution at the gene level. You have information also available about how do the isoforms look like. Now the mathematical part, which is common to all those three methods, and that's why I skipped that part for now, is how do you assign these partitions to, how do you assign these towns to the different partitions of these? So very simply, I think it's also there in my slide, so I might just use it. Okay, it's much easier to do it. So imagine you have three different isoforms, right? So you have three isoforms which are red, blue, and green. You get this data about, this is your genome, okay? These are your isoforms. So this is genome, which is fixed. These are three different isoforms coming from this gene, which is possible. You get 5 reads from your data. Read A, read B, read C, read D, and read E. The colors here also indicate how many, which transcripts does it, or which isoform does it belong to. Okay, so A overlaps with blue, red, and green. B overlaps with blue and green. C overlaps with blue and red. D overlaps with E. D overlaps with red, and E overlaps with green and red. Okay. Structure is clear. This, the aligner will tell you, black box aligner will tell you that this is what the mapping locations are. Now, I want you to tell me what is the quantification, or how many copies do you think are there in this data of isoform red, isoform green, and isoform blue? Is that problem clear? So the black box algorithm tells you 5 reads map in these locations. You have this information about this isoform because you have access to the isoform database, right? You know which isoform looks like what. So this is something which has been previously categorized. This is one possibility of an isoform. This is one other possibility of an isoform, and the red is another possibility of an isoform. Okay. Now, given this structure, I want you to assign probabilistically which read should be assigned to which isoform. Is the problem clear? Problem clear? Problem not clear? Jesh, problem clear? Okay. So you have this genome. Okay. This is, let's say this is gene X. Okay. Gene X has 3 possible isoforms. Exon 1, exon 2, exon 3. This is isoform 1. Isoform 2 has a mix of exon 1 and exon whatever, some other exon. and the last exon is common with respect to red. And then you have a blue isoform. So this is exon, this same exon structure as green, and then the middle exon, which is common with red. So three possibilities exist with respect to the isoform. Okay, you want to find out the reads. So the reads map, all the reads map to this gene. Okay, if you look at the actual coordinates, A maps has overlapping with red, green, and blue. B has overlaps with respect to blue and green. C has overlaps with red and blue. And E has overlaps with green and red. Okay. Okay. So five reads, but I have no idea. Is isoform red the most abundant? Is isoform green the most abundant, or isoform blue the most abundant? Okay, question here. Okay, how will you solve it? Okay, you begin with D, which is there only in E. Okay, so that gets assigned. Okay, very good. What about the others? So D is the simplest case, right? D only aligns to E. Sorry, red. This read, which is split, it aligns only to isoform E, isoform red, which means that at least one copy of isoform red should exist. That's how this read can originate. Otherwise, there's no possibility of the read originating. Okay. So let's say D, we can delete it. Doesn't matter. What about the other reads? How will you start? Like, what is the simplest assumption in these three isoforms? They are overlapping, so they are existing as copies. But what can you say something specific about what do you think their quantification could be? What is like the general, what is the simplest assumption in this case? Three isoforms. If I have to ask you on terms of relative abundance, let's say, so relative abundance, I will refer to, let's say, as alpha. So alpha R, alpha G, alpha blue. That sums up to 1. Okay, which means that on a relative scale, how many copies of each of these isoforms are there? This should be 1, right? Okay, this is the that we start. What is the simplest assumption for alpha RGB? All are. Who said? All are. You said. Somebody said all are equal, right? So alpha R, alpha G, alpha B, 1/3. Simplest assumption. Okay, now let's forget this problem for now. Okay, let's totally forget this. Let's do a point cost problem. I promise that I'll not bring point cost, but I thought I should bring it back. And you'll see why. So you have these 2 points. This is A, B. These are all biased. Theta A is, I don't know what it is. Theta A is the probability of getting a hex. Theta B is the probability of getting a hex. If we start with point B, I don't know what it is. Okay. I want you to do an experiment which will tell me what is theta A and what is theta B. What experiment would you do? Point. You have a bias point. You want to figure out what is the probability. How will you do it? You toss it, right? So let's say I do 10 tosses. I can do this for each. Okay, so if I do this for each, so let me take A. If I take A, maybe I get 5 head, 5 tails in first try. Then I do it again, I get something, let's say 4H, 16, third time, 5H, 5T, and so on. I can do this, let's say 5 times. Okay. How will I calculate theta A? So I did this 5 times. Assume it's a random number of tries. In each try, you're doing this 5 times. you're tossing the coin 10 times and you're doing 5 replicates of this strategy. How many? So what is theta A? Analytically, obviously. Analytically, what is theta A? No, it's not 0.5. I mean, okay, it could be 0.5. Good point. It should be 0.5. But analytically, it is just the number of heads that you did divided by the total, right? So this part here, both for theta A and theta B. So at this point, I knew that these had labels. A alag hai, B alag hai. And I can figure out what is theta A and what is theta B. Okay. Now, let's make the problem a bit more complicated, which is I don't know the identity of these. So I put my hand, I get a coin from this box. I have no idea which one is which. I can still do this experiment and I still want to find out what is theta A and what is theta. Or what are the two thetas, okay? You don't know the identity. So you still want to find out what is the probability of those two, individual probability of them getting heads. Okay, so you put this in a jar. Okay, you put this in a jar. So these are 2 points lying. They are both biased. There's theta A and theta B. I don't know which one is this. Right? And they look very identical for all practical things, but you know they are different. Like as in you know that these 2 values are going to be this. How would you tell me what is probability of, let's say, this point and this point? So the intuitive thing that we did here, which is we said the total number of heads divided by the total coin tosses, is also called maximum. This is also in this case the maximum likelihood estimator. I will not go into super details about it. I just want to give you the information. This estimate is what's called MLE. Okay, maximum likelihood estimates, which means that when you plug in the values of this, your likelihood function is maximized. Okay, and you know the likelihood function of this is given by theta A times number of heads 1 minus theta A times tails. When you plug in this particular value, theta A hat, this likelihood function, which is a function of theta that gets maximized, is called maximum likelihood estimate. Okay. We'll work. So there is a very good mathematical problem, very rigorous problem in some fashion. But just to sort of keep it intuitive enough for everyone, we'll just talk about it from an intuitive point of view. And the good thing about this problem is you can figure out the solutions intuitively without getting too deep in the maths. So I'll have a problem in the assignment. If you're interested in the math part of it, we'll go down that route. But just so that everyone is on the same page, I wanted to do the intuitive version here. Okay, 7 minutes. We should be able to do it. Okay, so I'm asking you this question, right? So in this case, when you had access to the identity of those points, you could toss both of them and you can figure out what is the right value of theta A and theta B. Okay. This is what this particular experiment is doing, and this estimate that you get is called the maximum likelihood estimate. So I took point B, I tossed it, I got 5 heads, 5 tails, 1. Then I took point A, I tossed it, I get 9 heads and 1 tails. I take it again, maybe I get 8 heads and 2 tails. At the end, I just tally the total number of heads about the total number of price, and that gives me the total value of the theta A. So overall, I get 24 heads for coin A and 9 heads for coin B. Okay, so that is the maximum likelihood. In this case, I knew which coin was being chosen when, and so I could do this separation. But if somebody blindfolds me, so you're blindfolded, these 2 points are in the jar, and then you take this out and you still are able to observe. The only thing you are able to observe is the heads and tails. this series you are able to observe. You're not able to observe what these values are. Okay, so you're not able to observe what is the identity of. How would you solve it? Sorry, say that again. You know the factor. Pattern. Okay, you know the pattern. Okay, pattern is a good. Yeah, pattern. These tosses are independent, so the pattern doesn't really. You're saying that the series of this thing, right? The series of HT that is that the pattern that you're talking about, or you're talking about the quantity? Quantity. Okay, okay. And how will you use it? Okay. How will you start this experiment? Sorry, I'll come to you, but you wanted to say something. You can keep track of the total. You can keep track of which coin you picked. That you know, obviously, in the sense that you don't know which is coming from where, but you know the identity that you pick coin 1 or coin 2. Okay, so let me clarify. Okay, so you put your hands in the jar. So you're choosing one coin. At that point, you don't know what happened. When you make the observations, right, you'll observe a series of heads and tails. You can calculate the probability whether it came from 1 or 2. That you can do. Like that you can separate. Does that make sense? No, okay, doesn't make sense. Okay. So you put your hand here, okay? Put your hand, take out any one of them. Okay. Let's say you get 5 heads, 5 tails. Now I don't know which coin it came from, but you're allowed to keep track of the tally. That what would happen if you knew something about 1, what would happen if you knew something about 2. That you're obviously allowed. Sorry if that was not. But does that help? So if you started with a presupposition, right? So if you started with this is let's say 1, this is let's say 2, and you assign them some probability, you can keep track of what is the likelihood of observing 5 heads and 5 tails if it were 1 or if it were 2. Is that clear?
Likely the opposite question. That is, given that I'm getting a head versus probability, it is coming from 1. That is, I use a base theorem here and.
Okay, very good. Very, very good. Okay, and then, okay, that's step 1. Very, very good. Okay, does everyone see which direction we are going into? So 2 more minutes, and we should be able to solve this in 2 minutes. So I will say that, okay, I have no idea, right? I have absolutely no idea what is theta 1, what is theta 2. But maybe it's fine. I don't care. Let's assume something. You can assume 0.5, but it's biased. So let's, again, they don't have to add up to 1, but I'm just giving you an example. Okay. So it could be any particular value that you start with. So the general solution that I'm going to just, maybe will not complete it, but just the general solution is a very beautiful mathematical or a statistical problem. It came from a signal processing paper by Dempster et al. The first time, again, this is one of those algorithms when I saw it first time, I was like, this just cannot work. This does not make any sense. But when you apply it and you do this as part of your exercise, you'll see that it does work and it works beautifully. It's applied in these general process problems, which is called the missing data problems, where you observe the final things that you observe, for example, the coin tosses, but you don't observe which coin it is coming from, but you're able to infer, and you're able to infer how these observations have arisen, or what is their probability distribution. So we can do what Aryan said. We don't observe this vector. Let's say this is Z. We don't observe the identity whether it's A or B, but I can always calculate once I observe this, what is the likelihood that this coin was A, given I see 5 heads, 5 tails, and the fact that I started from this assumption. This you can calculate at each step. Similarly, you can do P, C, D, right? So at each step, you can calculate what is the probability of observing it. So this is whatever, this is X, this is Y, your X is ZA, or it is the posterior, or what is the chance that you observe this to be A versus this to be B. You're observing the coin tosses, but you're not observing the identity, but you can still assign probability to them. What is the probability that it is either of these? Okay. So what do you do next? In terms of what we do next is we calculate once we calculate what is the likelihood of observing, let's say 5 heads and 5 tails, we can assign them, we can calculate the posterior. So the value that you're seeing here, 0.4, we'll do this by hand in the next class. Just stay with me for the intuition part, right? So we get the value about, once we calculate those posterior in the previous slide, this is what those posterior are. So I started with, this is 0.6 and 0.5. I get the posterior about what is the posterior probability that if I observe 5 heads, starting from this value, what is the probability that it was A versus what is the probability that this was B? Okay. Now you have these probabilities. What is the probability that it was A? What is the probability that it was B? You know that you observed 5 heads. You can break this pattern probabilistically. You can say that if I had 0.45 chance of picking an A and 0.55 chance of picking a B, I observed 5 heads. That means if this were to be true, then probabilistically, I would have observed how many heads coming from A? Chance of picking an A is 0.45. How many heads? You have 5 heads. Everything is happening probabilistically, which means that there is a chance of it coming from both of these in some part. You see 5 heads. A ka chance was picking was 0.45. So 5 into 0.45 of them likely came from A. Similarly, 5 into 0.55 of them likely came from B. Okay? And you keep repeating this experiment, and I'll talk about the next step in the next class. This, you will use it to re-estimate what is the probability of getting a heads from A and B, because at the end, you will again get a tally of how many heads are coming from A, how many heads are coming from B. You can recalculate theta A and theta B and do this continuously till this converges. And there's a beautiful theorem showing that this will converge. Will not again go there, but trust me that this converges, you get an actual estimate, which is actually very close to what the real value is. Okay. So this was a digression, but you'll see why this digression exists in this setting. Yes. No, it is not converging because of central limit theorem, but it is converging because the likelihood, the final likelihood equation that you get, it is the maxima there. So what you're basically saying is the maxima does exist, and that's realizable in terms of. So this happens for a particular class of problems. If your probability distribution which is generating that is fixed, this will always be. We need to be convex, exactly. That is the only requirement, and you can show that for a lot of these probability distribution that final estimate is actually convex. So you'll do this as an advanced bonus exercise. I will not talk about the actual math in class. So this is the same principle that is used to partition this, right? You don't know which began from where, but you can use the same principle to partition. So we'll do this exercise in the next class, and I'll talk about the actual methods before talking about other techniques to deal with this data. So just 30 more seconds. So once you get more data, once you get this data about D1, D2, D3, and get it across multiple samples, the first thing that you want to see is how similar is this data across different donors or not. So we'll talk about analytical methods to deal with it, PCA, SVD, dimensionality reduction, and so on in the next class. Okay, thank you. Thank you, and sorry for taking 5 minutes again.
This mean that
it could be either of these.
It could be either. You don't
That's
know which-
okay, right? So that is the transcript which is generated in the sequence, when it was sequence.
Right.
Okay.
Yeah, so you don't know exactly which came from where, and you would want to assign them. Sorry, one second, let me just stop this because it's recording.